Dear students, welcome to the fifth session of Molecular Basis of Inheritance. Let's learn about genetic code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires the transfer of genetic information from a polymer of nucleotides to a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There existed ample evidences to support the notion that the change in nucleic acids were responsible for change in amino acids in proteins. This led to the proposition of a genetic code that could direct the sequence of amino acids during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and deciphering of genetic code were most challenging. In a very true sense, it required involvement of scientists from several disciplines like physicists, organic chemists, biochemists, and geneticists. It was George Gamow, a physicist, who argued that since there are only four bases and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made of three nucleotides. This was a very bold proposition because a permutation combination of 4 raised to 3 would generate 64 codons, generating many more codons than required. Providing proof that codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combinations of bases. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severo Ochoa enzyme was helpful in polymerizing RNA with defined sequences in a template-dependent manner. Finally, a checkerboard for genetic code was prepared, which is given here in the table. Let's have a look into salient features of genetic code. The codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acids, hence they function as stop codons. One codon codes for only one amino acid, hence it is unambiguous and specific. Some amino acids are coded by more than one codon, hence the code is degenerate. The codon is read in mRNA in a contiguous manner, hence there are no punctuations. The code is nearly universal. For example, from bacteria to human, UUU codon would code for phenylalanine. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. AUG has dual functions. It codes for methionine and it also acts as initiator codon. Let's look into the mutation and genetic code. The relationships between genes and DNA are best understood by mutation studies. Effects of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutation will be explained here. A classical example of point mutation is a change of single base pair in the gene for beta globin chain that results in the change of amino acid residue glutamate to valine. It results into a diseased condition called as sickle cell anemia. Effective point mutations that inserts or deletes a base in structural gene can be better understood by following the simple example given here. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. Insertion or deletion of three or its multiple bases insert or delete one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered from that point onwards. Such mutations are referred to as frame shift insertion or deletion mutations. This forms the genetic basis of proof that codon is triplet and it is read in a contiguous manner. In this session, you have learned about genetic code, its salient features, and mutations. With this, we come to the end of today's session. Thank you and have a nice day.